but I'm also an assistant lecturer from Muhimbili University of Health and Allied Sciences. I'm going to give a talk on association between genetic variants and F cells in sickle cell disease. Tanzania is populated with over 58 million people, of which children who are born with sickle cell disease per year are estimated to be 11,000 babies, and deaths due to sickle cell disease of below 5 years are estimated to be around 1,500,000. And currently in Tanzania, there's only one drug that is available for treatment of sickle cell disease. Hence, there is a need for research to understand the mechanisms of disease in order to develop new interventions. So sickle cell disease was actually the first human monogenic disorder to be characterized at the molecular level, and this is just a schematic diagram that illustrates how the point mutation occurs in sickle cell disease. So at the gene of the beta globin gene, at the 6th position where you have adenine, there is a point mutation at this position. Instead of having adenine, you end up with thymine, and as a result, instead of having glutamic acid amino acid, you end up with the valine amino acid. This produces a mutant globin protein, and the glutamic acid is actually an hydrophilic amino acid, and valine is an hydrophobic amino acid. So the abnormal hemoglobin in which valine has replaced glutamic acid causes the hemoglobin to be less soluble under hypoxic condition, and therefore this hemoglobin they do polymerize into crystals that distort the red blood cells into sickle shape. As you can see here, these patients tend to have these sickle shapes. They tend to have these sickle cells, and these sickle cells do block the blood flow in the blood vessels, and as a result, these patients tend to have pain in the joints, and actually this is one of the pathophysiology of the sickle cell disease. So fetal hemoglobin, which is the predominant hemoglobin at the gestation age of 40 up to the time when the babies are 2 years, is one of the major modifiers of sickle cell disease severity. And this is just in an illustration that shows the erythropoiesis process that takes place in the normal individual and in the sickle cell disease individual on the right. So on the left, this is the marrow of a normal individual, whereby there is a gene switch from gamma to beta, and as we know that the normal adults normally have higher percentages of adult hemoglobin variant as compared to other hemoglobin variants. So during the gene switch, the switching is not so complete in such a way that we tend to retain some small amount of fetal hemoglobin within our red cells, and this fetal hemoglobin is retained in a subset of cells known as F cells. So a normal individual has below 1% of fetal hemoglobin and below 7% of F cells, and majority of the hemoglobin is the adult hemoglobin. Whereas in sickle cell disease individual, we know that these individuals do undergo hemolysis. So in order for the bone marrow to compensate for the loss of these red blood cells, it undergoes stress erythropoiesis, and as a result, there's accelerated erythropoiesis, and there's an increased levels of fetal hemoglobin being produced, and subsequently higher levels of F cells will be present in these individuals with sickle cell disease. So the levels of fetal hemoglobin and HbF is not stable within in these individuals. There's a high variation amongst these individuals with sickle cell disease. There are many factors that influence fetal hemoglobin and F cell levels that influences this variation that is seen in sickle cell disease individuals, and some of these factors include age, sex, the alpha globin gene, the alpha globin gene number, the X-linked locus, as well as preferential survival of F cells in peripheral blood. And the number of F cells in sickle cell disease patients actually depends on two main factors. One is the increase in proportion of erythroid cells that still make the gamma chains as a result of stress erythropoiesis, as we've seen in the previous slide. but also the preferential survival of F cells in the peripheral blood because of their relative resistance to sickling. As we know that these F cells do not undergo the polymerization process due to the tetramer structure of the fetal hemoglobin. So as we've mentioned, there are many factors that influence the variation that is seen in sickle cell disease individuals. Genetic factors are one of the major factors that influence this variation. And previous studies have looked into these genetic factors, and they've identified 3 known loci in chromosome 2, 6, and 11. However, these loci only explain about 20% of the variation of these fetal hemoglobin and F cells that are seen in most of the sickle cell disease patients of African ancestry. Henceforth, there is a need for further studies to investigate this missing heritability of fetal hemoglobin and F cells in sickle cell disease. And hence, in our study, we aimed to investigate how F cell percentages rather than fetal hemoglobin correlate with their polymorphic genetic determinants. Our study was actually carried out at the sickle cell disease clinics at Mwananyamala Temeke na Mana in Dar es Salaam, Tanzania. And this included, our study design was retrospective and cross-sectional study. And our study population were actually sickle cell disease individuals that were attending the sickle cell disease clinics. We used convenience sampling to select for the study participants. In order to do the statistical analysis, we used Stata version 14 as well as the AfSHG. For descriptive statistics, we used median and interquartile ranges for the continuous variables. And in order to test for gender differences, we used the Wilcoxon rank sum test. To determine the effect of different genetic variants on F cells and HbF, we used the univariate regression model. So this is just a flowchart that illustrates how the sample size was generated. So my colleague and mentor, Dr. Siana, had generated data of about 2,000 individuals. So she conducted a study, actually it was one of the largest genome-wide association study of sickle cell disease individuals in Africa. And so we have data of these about 2,000 individuals, of which out of these 1,400 had HbF level measurements. From this 1,400, those that were above, those that were 5 years and above were about 1,200, and from this we're able to recall by using the convenience sampling, and we ended up with the 227 patients. However, 5 of these individuals did have contradicting HbF levels, and therefore we ended up with a sample size which was 222. So as I said, we wanted to evaluate the association between genetic variants and F cells. First, enumerate the F cells, and we use the Fax caliber machine. As you can see in this picture, this is a machine that we have in our university, and it's a 4-color machine. And before acquiring the data, we had to first fix the cells using the glutaraldehyde, and then we had to permeabilize using Triton X, and finally stain with the anti-HbF. After this, we acquired the data in the machine, and we did the analysis using the FlowJo software. And this are just the results from the FlowJo, where the results can be presented either in the dot plot or in the histogram format. And for this particular individual has about 42.3% of the F cells. And looking at our findings, we did look at the gender differences in hematological parameters of the study participants, and we observed a statistically significant difference between males and females in all of the red cell indices except for hemoglobin and MCHC, but we also found there was no statistically significant difference in the white blood cells count as well as the platelets counts. We also observed a different distribution of F cell percentages in females compared to males, and this was actually similar to a study by Creel et al. In 2009. And this fact actually led us to hypothesize that some or most of the differences that we've seen in the previous table may be a consequence of gender difference in F cells percentages. So we went further to investigate the X-linked XP22.2, which is the F cell production locus, which was previously studied. So the conspicuous difference in F cell percentages distribution between males and females was actually suggestive of an X-linked genetic determinant. So this locus was actually first studied by Dover et al. In 1992 through a pedigree analysis in some families with sickle cell disease, and this led them to postulate that an X chromosomal dominant genetic determinant might be on the XP22.2. So we tested our variable, which was fetal hemoglobin and F cells, and we treated this with as continuous variable, and we used a candidate SNP approach. We picked the two candidate SNPs from studies by Soloviev et al. In 2010 and Chang et al. In 1995. And these two SNPs reside in the FRMPD4 candidate gene. And when we did this association, when we did this test, we observed that there was no association. We then tested the association of F cells separately between males and females using arbitrary cutoff values in F cells of 20% in males and 40% in females. We found a significant association with the RS16998911 that is found at this region within the 5' untranslated region of FRMPD4 in males with high F cells. So since 20% cutoff value was arbitrary, we went further to explore different cutoff values both in females and males, and we can clearly see that in females across all the F cell percentages, we see that there is no significant association. However, in males, we were able to see an association to be robust over a range from 13% of F cells up to 30% of F cells. We also scanned the chromosomal region of XP22.2, which comprises of about over 4,000 SNPs, and we observed that 3 SNPs in males and 1 SNP in females were above P value of 10 to the power of -4 bar, which tagged the FRMPD4, and adjacent to this was the MSL3 and the TL8 genes. However, a signal that reached the threshold of GWAS significance, which is 10 to the power of -6, was confirmed in males for HbF percentages against the RS16998911, and this was actually the only signal amongst the 4 stated in the previously to have reached the criteria of significance. So apart from testing the FRMPD4 gene with the candidate SNPs, we were also interested to look at whether the previously known loci were associating with variables from our population, and we found that from the 3 on chromosome 2, 6, and 11, we found that there was a statistically significant association in BCL11A, specifically the SNP of RS11886868 in HbF levels and F cells. However, we did not see this significance in chromosome 6 and 11. And as I've explained in the previous slide, we're able to see the significance in males with high HbF as well as F cells with high, males with high F cells in these individuals. However, we're not able to see the same for the females. So to conclude our study, given the gender difference in distribution of F cells in sickle cell disease patients, We have studied the genetic association after gender sorting, and analyzing males and females separately may actually be key to finding associations for X-linked genes. This has yielded a positive result for fine mapping of long-sought X-linked determinant of F-cell percentages, and these findings were actually validated. Our study has also highlighted the need for deep sequencing of targeted loci that might not have reached the GWAS significance in the previous studies. We can strongly now conclude by saying that apart from the three known loci that modify, apart from the three modifying genes that have been previously reported to play a role in modulating the levels of fetal hemoglobin and F cells, we have now added a fourth candidate gene that may play a role in modulating the levels of fetal hemoglobin and F cells in sickle cell disease individuals. On the future follow-up studies, we hope to do replication studies in additional sickle cell disease population with larger sample size, and also we plan to make use of or benefit from the multi-country consortium which is created by Sickle in Africa and the H3 Africa in order to design the GWAS array and bioinformatic analytic tools which will account for the African genetic diversity. We also plan to design a study to understand the mechanisms whereby a gene on the XP22.2 affect the gamma to beta switch and therefore the number of F cells. By this we mean that we plan to do these functional studies using the erythroid cells in order to understand better the mechanisms of this particular gene. This work was published last year in October in the British Journal of Hematology. Lastly, I would like to acknowledge the study participants, the staff of MUHAS, specifically from the Department of Biochemistry and Department of Hematology and Blood Transfusion, my collaborators from King's College London, specifically the Red Cell Group, the funders of this project, the MUHAS CEDAR Small Grant, as well as the Commonwealth Scholarship Split Site, and my supervisors and mentors and all the co-authors of the paper for this project. Thank you.